Hi, everyone, and welcome to Research America's webinar today. Most importantly, Happy New Year, if wishes can come true. And thanks so much for joining us and for your partnership as always. If your organization is not a Research America member, that could be a New Year's resolution. Get in touch with us, get in touch with me. I'll connect you with the right folks and join our alliance. I think it's going to be a year where advocacy is so important, as it is every year, but we have a lot of challenges and we need to overcome them in the policy and advocacy arena. So join us. I am so thrilled that our first special guest this year is Dr. Eric Green, who is the director of the National Human Genome Research Institute. I'm sure you all know him. Dr. Green, who is an extraordinary leader. He played a pivotal role in unraveling the human genome and is going to speak with us today about the role researchers supported by the National Human Genome Research Institute. And I'm spelling it out because I'm afraid that I would get the acronym wrong, plays in advancing medical and public health progress, and his vision looking forward for the Institute and for genomics. As always, please type your questions into the Q&A box or chat. We'll do our best to pose all of them when we get to the Q&A portion of today's session. With that, let me turn it over to you, Dr. Green.
Well, thank you, Ellie. Happy New Year to you. Happy New Year to Research America, and Happy New Year to all those who were kind enough to join us today. I'm delighted to be able to provide an opportunity to discuss some exciting developments in this field of genomics that I've been involved in, but most importantly, to tell you about what we're anticipating for the future. And so I am going to give a very high-level overview of some points I want to make, and then I will stick around as long as you want to answer as many questions as you have. I would just start off by reminding you that this discipline of genomics is a relatively young discipline. The word genomics was coined in 1987. The field was sort of named genomics in this lead editorial of a new journal that was set up at the time called Genomics. And it really grew out of the molecular biology revolution of the 70s and 80s that really provided laboratory scientists some of the earliest methods for cloning and manipulating and isolating DNA, and importantly, for reading the building blocks of DNA, as we abbreviate them, G's, A's, T's, and C's, the sequence of DNA, that gave us these abilities to begin to contemplate the idea of analyzing all the DNA of an organism, which is called its genome, and doing so perhaps in a really big, audacious way. So the field was named in 1987 really because of the idea that something incredible was going to happen in the biological sciences. And that incredible thing was the Human Genome Project, an effort that was internationally supported through a very large consortium of scientists, multiple countries. I had just graduated medical school and graduate school and got involved on day one of the Genome Project as a trainee, and dedicated my career to genomics ever since. The project is, now in our rearview mirror, and we embrace it as one of the most successful endeavors in science, both because it accomplished all of its goals, including sequencing the human genome for the very first time, but also because it set up a trajectory of accomplishments of this new field of genomics that are catapulting us into the future, as I'm going to describe. And I really like to remind you that, we're now only in our 3+ decades of genomics, now, sort of still not even 40 years of genomics. We have really accomplished a tremendous amount. I told you about how the first highlight of genomics really was, I think, in many ways, the generating that first sequence of the human genome by the Human Genome Project. But it's really important to recognize that, 2003 is now 18 years ago. We certainly didn't stop there. That was not going to be the finish line for genomics. That was going to be the starting line in many ways. And one of the first things we needed to do was to reduce the cost of actually sequencing a human genome, because that very first human genome sequence cost us about $1 billion. One day we wanted to see the ability to sequence a human genome at a cost approaching a clinical test, for example, so that we could use genomics in medicine. I can now tell you very proudly that in the last 18 years, my field of genomics has developed technologies that have reduced the cost of sequencing a human genome more than 1 million-fold. It now costs less than $1,000 to sequence a human genome. And as a result, we are now seeing it used in all sorts of ways including in the practice of medicine. Having developed very inexpensive methods for sequencing DNA and sequencing genomes, what it meant is we could have a third highlight on this slide, because we have now gone from sequencing one human genome 18 years ago to now sequencing millions of human genomes. And we don't do this just for sport. We do this because we want to compare people's genomes, and we want to catalog the roughly 1 in 1,000 letters that are different between any two people's genomes. We differ by about 0.1%. We're 99.9% identical at our sequence level, but that 0.1% has within it, and each of us carries millions of these spelling differences, a small fraction of which can turn out to be medically valuable to know, because we believe it's going to point to better ways of practicing medicine by having unique genomic information about individuals. Now, in order to do that, we need to understand how those variants, how those different spellings in our DNA affect genome function. And we have come a long way in understanding how the letters, the 3 billion letters that make up the human genome, actually encode the information for developing and operating a human being. And we then are learning increasingly how spelling differences in some of those sequences influence our health and disease. So it's that interplay of how we differ and how our genomes each uniquely work that really have a lot of influence on health and disease. And we've taken that growing knowledge and have greatly advanced our understanding about the genomic bases of human disease, rare diseases and more common diseases. And it turns out that almost every human disease has at least a genomic influence, if not an overt genomic cause. For rare diseases like sickle cell anemia and Huntington's disease and cystic fibrosis, it's often just one gene in the human genome that is mutated, has a spelling change that breaks that gene. For more common diseases that fill hospitals and clinics around the world, like hypertension and diabetes and differential susceptibility to COVID-19, those things are complicated because it's not one gene, it's an interplay of multiple genes, different spellings of multiple genes, what is often a greater influence of environmental and social factors. We will spend a considerable time in the next decade or two completely unraveling the genomic basis of rare and common diseases, but we've made incredible progress, especially for rare diseases, because they're simpler to understand, simpler to diagnose. You're looking for the breaking of one gene in most cases. And as a result of these first 5 accomplishments, I'm incredibly proud to tell you, we have vivid examples of genomic medicine now emerging. Genomic medicine being using genomic information as the part of the tailoring of medical care. We are now increasingly using the sequencing of a patient's genome as part of their medical care. It's the earliest examples. It's happening mostly so far in cancer and rare disease diagnostics and in prenatal testing. But increasingly, I think you'll see it more and more if we make the kind of achievements I think we're going to make become more mainstream in the coming decade or two. That's the highlight reel, and I realize it's all very high level, but I really want to emphasize now how we really got there. And I don't want you to think just because I have really slick, nice, pretty looking PowerPoint slides that any of this in the past 3+ decades of genomics has been easy. This has all been research. It's all been tedious, difficult research. It's all been a journey, and it has never been linear. Like all other avenues of research, it has many twists and turns, and we are constantly adapting our strategies to achieve the next set of goals we have in front of us. And I really want to emphasize that these twists and turns have been guided by strategic input from the research community, allowing us to every once in a while take stock of what we're doing and then retooling and then reprojecting a new set of objectives going forward. If we think about the trajectory of genomics over the last 3+ decades, I can tell you that the Human Genome Project was guided by 3 strategic visions that the international group of scientists involved in the Genome Project put together to guide us along the way to be successful in the 13-year mission. Our institute, NHGRI, the National Human Genome Research Institute, which I've been at now for almost 28 years, I've been the director for the last 12+ years, when the Genome Project ended, put out a strategic vision that guided us through the end of 2010, and then we put out a new strategic vision that guided us through the end of last decade. But as we entered the new decade last year, and when I think about a year ago, New Year's, and we brought in the new decade in particular of 2021, we recognized we wanted a new strategic vision to guide us for this coming decade. And we also realized that Genomics once was quite isolated just to the people involved working in genomics, like people working at my institute or people funded by my institute. But one of the key lessons I really want you to take away from my presentation is that genomics is everywhere now. Genomics once upon a time was really just about the Genome Project and putting a magnifying glass on the human genome. But the threads of genomics over the last 18 years, since the end of the Human Genome Project, have increasingly been woven into the tapestry of biomedical research. Basic research, model organism research, bench research, more recently into human genetics and data science and bioinformatics, and then in particularly over the last 6 to 8 years, increasingly into patient care and into many other areas of medicine and society and the whole research enterprise. And as we look to the future, more and more diverse disciplines involved not only in research and in medicine and society will continually be weaving in the threads of genomics into their work. And genomics has become pervasive and highly disseminated across the entire biomedical research enterprise. And so when we went to put together our latest strategic vision, which is shown here, which was published in 2020, about 13 months ago, which was supposed to be a blueprint for the future of genomics in the coming decade, we recognized it wasn't just about coming up with a vision for genomics, for the genomics community, but rather it was about coming up with a vision for how genomics was going to operate at the forefront of activities to facilitate all of biomedical research as we saw genomics get implemented into medicine and as we saw genomics become more mainstream in society. I strongly encourage you to look at this paper. You can come to this URL, you can get it for free, download it. It's 10 pages in Nature. It is filled with an incredibly dense amount of valuable insights about the future of genomics that we hope will play out in a very positive way over the coming 10 years. Very briefly, the other thing I will tell you about our new strategic vision in particular, which differed from the previous ones, was the heterogeneous elements that we put together in forming the vision. It wasn't all about big research projects, and it wasn't even all about research per se. In fact, at the end of the day, our strategic vision, which I hope you will read, captures elements at the forefront of genomics that fall into 4 major categories. One of the categories relates to important values and principles that undergird the field: collaboration, data sharing, standards for data quality, so on and so forth, diversity, equity, and so on. And so we articulate what those principles and values are that are pervasive and important that undergird the whole field. Part of what we heard was the importance at the forefront of genomics of maintaining and enhancing a foundation for genomics that everybody would benefit from. Because if everybody's using genomics, we need to make sure everybody has people trained in genomics, technologies for using genomics, the means by which data is shared through data resources, and so on and so forth. We articulate a series of foundational elements of genomics. We also articulate the set of barriers that exist going forward. 18 years ago, the big barrier, cost of sequencing a genome. Now, a millionfold reduction, that's not such a barrier anymore. There are other barriers. We describe some of those barriers and how important those barriers are to now knock down so that everybody can surge ahead in using genomics for their particular applications. And then finally, of course, we describe compelling research projects. We always have big, audacious projects to do in genomics, and 2020s are no different than any other decade in genomics. We describe some of those, and all these together sort of all meld together to represent what we think is the forefront of genomics for the coming decade. The other thing we did, which is what I want to sort of end on because I think it really is a very upbeat thing to think about, is unlike our previous strategic visions, we chose this time to add a new component to our strategic vision. We set out a set of 10 bold predictions about what we think might happen in human genomics in the coming decade. And we did this to be deliberately audacious, to be inspirational and aspirational. I want to be upfront. We don't expect these 10 predictions to all come true. If even 1 or 2 of them came true, we'd be incredibly successful. But we thought it would be valuable to lay out the kinds of things we see out there, way out there, maybe more than 10 years away, but the things to strive for, the things to reach for, the things to aspire to. We put a short video together that we rolled out when we published our strategic vision. So I want to share this with you now because I think it sort of shows the breadth of ideas we have about what genomics might bring us in the next decade, or maybe slightly beyond for the harder, bold predictions. So see what you think.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future, a future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2. The role of every gene in the human genome will be known. Prediction 3. Environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4. Genomics will no longer use social constructs such as race in human research studies. Prediction 5. Student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6. Genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7. It will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8. A person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9. Advances from human genomics will benefit all of society and not just a few. Prediction 10. Genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
Well, I hope you enjoyed that video. One of the core values we have at NHGRI, and my communication team very much embraces this, is that because we are at the forefront of so many exciting developments in science, in medicine, and society, we feel strongly that the value and the importance of communication and sharing information. And so we actually recently created we have so many electronic resources to share with interested people. And if anything I talked about today you find interesting, I'm sure you could find a lot more if you wanted to dig deeper. So we created this one-stop shop. It's really easy to remember, genome.gov/stay connected. And when you go there, you'll see this website. But if you just scroll down, you will find all sorts of opportunities to dig deeper. First, our website in general, genome.gov, is incredibly rich with a lot of information. You can stay connected with me specifically, either through a monthly newsletter or through following me on Twitter, which I have a very active Twitter account. We also have the strategic vision that you just heard about, as well as a brochure about the Institute. And meanwhile, we're very video oriented, and we put a lot of materials in meetings and seminars and all sorts of series on our Genome TV channel of YouTube, as well as capturing a lot of oral histories of people in the field of genomics. If you're interested in those, you'd find a wealth of information if you just keep digging into this site. And then also, if you scroll down further in the site, if you want to hear a longer version of a talk from me, we put some talks that are a little more lengthy, a little more detailed, a little lower altitude than what I described to you today. You're welcome to look at those. If you want to listen to podcasts or read op-eds that I've written or I've participated in, those are also available on the site. So I'm going to stop there. Pretty much exactly the time I wanted to do, and would love to hear what you have on your mind, answer any questions you have, and let's take it from there. And I'll stop sharing my screen. So
Thanks so much, Dr. Green. That was terrific. A lot to take in and think about. I want to start with a question that is going to belie my ignorance, but can you describe the difference between genetic testing and genomic testing?
They're really, or I could even back up. What's the difference between genetics and genomics? And there, in many ways, they get sort of intermixed. I mean, let me start with that my definition. And by the way, if you ask 6 other geneticists or geneticists, they'll tell you a slightly different approach of how they parse those two. Genetics is the study of inheritance, the study of traits that go from one generation to the next. It turns out all that takes place with DNA, and genomics is the physical study of DNA. And so genomics tends to be more oriented around the comprehensive analysis of the DNA molecule, and genetics is much more attentive to inheritance patterns, because it really is about inheritance, and of course, the genome is the vehicle by which inheritance happens.
Right. So it's much more about risk factors versus what you got, what's happening right now.
I think genetic testing and genomic testing are now used interchangeably. In either case, you're almost always doing some sort of study of DNA. One difference might be you'll start hearing more and more about genome sequencing, or sometimes you'll hear the phrase exome sequencing. Now I'm getting geeky. Exome sequencing, exons are the parts of our genome. It's only 1.5% of our genome that directly code for protein. They're protein coding genes. So a shortcut is, and really inexpensive, is you could do an exome sequence, which is basically the 1.5% most currently known meaningful part of the genome. Those are really genomic tests because they're being comprehensive. But sometimes people say, well, I'm getting genetic testing. I mean, the testing phrase, they're probably interchangeable. But increasingly, all genetic testing is going to be done by genome sequencing.
I want to move to something a little bit tougher. And you shared with us your strategic vision. A couple of questions have come in from Lisa Schlager they're very good, but they're tough. And I've been wondering about it a little too. You mentioned that incredibly, the cost of genomic testing, you've been able to reduce it by, you said 1,000,000%, basically. It's down to about 1,000, under 1,000.
It went from 1 billion to less than 1,000.
Less than 1,000. So for those who don't have insurance coverage, for example, that's still big spending. I think about it in particular with rare diseases and the odyssey parents go through trying to come up with a diagnosis, when, as you've said, a lot of rare diseases is one, at least you can find out that it's associated with one, problem. In your strategic vision, is there any is there any room for those kind of issues and also privacy issues and that kind of thing that goes to maybe the impact of having sequencing on your ability to be eligible for certain types of insurance, that kind of thing?
So, so all of these things are within the purview of our responsibilities and certainly are touched on in the strategic vision. And let's just take each of these separately. The payment, and this falls in with the health equity, set of health equity.
Yes.
I would actually, so this is of great concern to me about, won't it be tragic if genomics develops all these incredibly valuable opportunities, but even $1,000 is too expensive. That would be, although that starts to be the cost of an MRI, which is largely perceived as a routine test now in medicine. But
that's something
what I would actually say is that in the case of rare diseases, and as you described, a diagnostic odyssey, we've actually gotten the most traction there because one of the responsibilities we have as an institute is to The payers say, generate data to show me that this will be bring more effective healthcare, and of course will be cost effective. And we've actually gotten probably more traction for individuals on diagnostic odysseys with rare disease than anywhere else. And because we've demonstrated that when you have an undiagnosed condition, you could burn through a considerable amount of healthcare and diagnostic testing that makes $1,000 look like a bargain. And as a result, and there's many settings for this. By the way, one of the really exciting settings where this is being implemented more and more is in a neonatal intensive care unit. You have a newborn child, don't know what's wrong with them. That child is doing very poorly. We now have the ability to sequence and analyze their genome in less than 24 hours. This was a major set of projects we supported that showed this could be an effective way to not only save babies' lives if you can figure out what's wrong with them and intervene, but in the long run, save a lot of money by not having them have prolonged stays in a neonatal intensive care unit if you can get therapy on board earlier. So that there are many healthcare systems that will now pay for that. And there's federal legislation that's percolating through Congress that might make that a more national circumstance. But I wanted to just,
I just want to mention real quick, if one of our folks wouldn't mind putting that legislation, I think it's called the Make Genetic and Genomic Testing More Accessible Act. If you can find that and put it in the chat, it's so relevant to this discussion.
And I would also point out some healthcare systems have voluntarily said, all right, we'll start doing that for undiagnosed conditions. Then you raised issues about privacy, and there's a whole other set of issues, but we certainly completely recognize that like many aspects of our lives, there are privacy concerns associated with genomics, just like our health information, just like our financial information. And just as we need to build as robust security protections for any of the information about us, we need to be doing the same thing in genomics. And that also needs to be met with stronger laws and policies and protections that all of us deserve to make sure that any private information about us is not misused. The good news, the progress we've made is having a Genetic Information Non-Discrimination Act, which gets us part of the way there in terms of preventing genetic discrimination. It probably needs to be built upon, and we have efforts to try to do that. But certainly, we should never take our eye off the ball and think that privacy is not going to be an issue, and we need to be constantly aware of it and do everything we can to be as protective as we can of that information.
It's terrific, and there's some really great comments in the chat that build on that. And why don't you join us, because I have approximately 3 million questions I could ask, but it's actually time to turn to the many great questions that we have coming in.
Yes, absolutely. So one question that has come in, Dr. Green, well, the person first says, great presentation, thank you so much, and sentiments that have been echoed. But can you give us an example from the strategic plan of a barrier that you're working to break down, and perhaps something that non-scientists can help accomplish?
The second one's hard. I mean, the first one that comes to mind, I mean, there's a number of barriers. Let me give you two different types of examples. One area, and this is a little geeky, a little hardcore science, but I can't hide from it, is, sometimes we represent ourselves as a single individual with one genome. Half of which we got from mom and half of which we got from dad. And it turns out we actually are more complicated than that, in that along the way, in the trillions of cells in our body, some of them, as we age, they pick up DNA changes, so that we have what's called mosaicism. And there's even some human diseases that are caused by people who pick up diseases from a subset of their cells that are mosaics. So one of the big barriers, but that's really hard. So like when you say, I want to study Eric Green's genome, it's one thing to draw some blood for me or take a cheek swab and sequence my genome. And that's sort of my average genome, because that's what 99.999% of my cells have as their genome. The problem is, of course, you could look at subset of my cells that might be telling you a slightly different story. But just imagine the complexity of that. So it sounds almost science fiction, but we really do need to have ways of being able to study mosaicism in a much more robust way, because we are learning that there are diseases out there that it's because of mosaic processes that lead to the
Wow.
disease. Now, I'll give you another example of barrier is we fully appreciate that genomics is not everything, that genomics is just one piece of the puzzle of why we are healthy or why we are sick. And the only reason why we talk a lot about genomics is that, well, first of all, that's what we do. And then we're riding this incredible technology surge of we can get a lot of genomic information on people really inexpensively. So we generate a lot of that data. But of course we realize there's lots of other things in our lives that affect our health and disease. I mean, there's infectious agents, but there's, as we're all living through now, but there's, there's our social context, there's our environment, there's, lots of things we're exposed to. And so you're increasingly hearing about other omics, like genomics. Well, then there's other omics. There's other parts of our life. There's other aspects of it. And as technologies get better, and we think about Fitbits as getting lots of physiological data or lots of sleep data or exercise data, as you imagine those massive data sets coming in, of course, you want to integrate them with genomic data. And it's another mind exploding ideas. Imagine if we can just take in all of this data about ourselves, and as new technologies become available, we're going to want to have multi-omic studies of people.
Right.
And that'll give us insights. And you'll quickly say, oh my gosh, we're going to need artificial intelligence. Yes. We're going to need machine learning. Yes. We're going to need, now here's where people can come in. You said, how can people help? We need the next generation, all these kids in grade school, middle school, high school, college. The more they can learn data science, we're going to need an army of really bright data scientists, because increasingly biomedical research is going to become a data science enterprise. And so one thing we can all do is let's train the next generation. Among those who are getting trained in various ways, let's get them really excited and get them cross-trained in biology and data science, and they will be put to work and they'll make the discoveries of the 2030s and the 2040s.
Great.
That's great. Thank you. So another question that has come in, this person is asking if it would be possible for you to provide an update, Dr. Green, on the status of the NIH All of Us national cohort that's being formed. And the person's also asking if that's under the auspices of your institute. And they're also curious about your thoughts, on the contributions of the overall effort.
Yes, so what first of all, great questions. Love to talk about All of Us. For those who don't know what All of Us is, it is a national program that President Obama launched and actually it just relates to what I was just talking about. So I really like the timing of that question, where basically 1 million Americans are being enrolled in a long-term study where they've agreed, as they're being enrolled, to share lots of information about themselves in a very secure way. It'll include their genome sequences. It'll include their electronic health records, massive amounts of data on all of us, on all people with our electronic health records increasing. And it'll involve capturing a lot of other data about them, including exposures, lifestyle, et cetera, and doing so for probably several decades, I would expect. And this is called a cohort study. It's not the only one going on in the world. There's other major ones going on in the world. The big successful one so far in particular is the UK Biobank, and which are 500,000, I think, people from the UK, and it's proving to be a very fertile set of data for doing lots of exciting science. We love this program because it very much was built on our belief that genomics was a starting point, but we wanted to build on other omics, including and other data such as electronic health record data, and intertwine those different data sets to be able to tease out the different contributions to health and disease, some of which will be genomics, some of which will be lifestyle, but doing this all in the context of a tremendous amount of medical information about people, what their diseases they have, what their blood counts are, what their other biochemistries are, what medicines they take, what medicines they do well on, what medicines they don't do well on. All of that is a fertile ground for, in the long run, being able to practice precision medicine. I've talked about genomic medicine. Precision medicine is taking all aspects of somebody's life and being more precise in tailoring their medical care. All of us
I'm sorry to interrupt.
do exactly that. It is not run by my institute. We are very partnered with them in very major ways. It is run out of the office of the director, and it's run by a gentleman named Josh Denny, who's a good friend and colleague. And where are they? They are still in the enrollment phase. I don't know their exact numbers, but I think they're well over 500,000, I believe. Unfortunately, slowed down by the pandemic. Very hard to enroll people during a pandemic, as you can imagine. But they're getting their momentum back. Data starting to come out of it. A lot of genomic data being generated.
Wonderful.
I would certainly tell you that over the next 10 years, you're going to hear a lot about All of Us studies being done using All of Us research data.
And Dr. Green, let me interrupt Anna for 1 second. You've mentioned, and I think that it's really great to hear that you're thinking broadly about the factors that influence health. But there is, in terms of social determinants of health, the direct factors on human in gene expression. Ali asked a question in our chat about that. Is there research ongoing about that in terms of the social determinants of health?
Yeah, no, sometimes people there absolutely is a subset of that. People often talk about, they classify sometimes as sort of gene environment interactions, and of which social determinants are one aspects of environment. There are other environmental aspects, and there's absolutely no question. In fact, there's actually a major workshop next week that my institute's doing with another institute at NIH that focuses on environmental health, and to exactly look at that. But All of Us research program, which we were just talking about, of course, is collecting a lot of data about social determinants, and that data will be able to be integrated with genomic data, electronic medical record data, and there's a high expectation that a lot will be learned by data sets like that, because it will be the first time you have all that data available to you to really hardcore look at what are the genomic contributions, what are the social determinant contributions. And it's another reason why, I didn't mention this before, but many of you probably know this, that All of Us has, as part of a signature aspect of it, is to over-recruit individuals who have traditionally been underrepresented in biomedical research. In other words, too much of biomedical research in the United States has been mostly people, Caucasians, but I think it's like 70 or 80% of the people being recruited, and also it's Caucasian, it's also people often in big cities near major medical centers, et cetera, et cetera. It wasn't very diverse socioeconomically, geographically, or ancestral origin. All of Us is bending over backwards to do just the opposite. So we'll not only have 1 million people, we'll have 1 million people, including mostly people who have traditionally not been part of biomedical research by all the things I just named. And that will also provide very fertile data that's been previously unexplored in the United States.
Dr. Green, that brings up a question that Annetta Schmidt asked, I think is really, I agree with her about being excited about what you said about the race as a construct.
Yeah.
That the role of genomics in really, in one of your bold predictions, that race will no longer be the same kind of a construct in research and hopefully more generally. But does what you're talking about with All of Us factor into that?
Well, correct. I mean, and in other words, it's exactly what I said, is there's so many different dimensions of diversity. Self-identified race is just one, but we also, All of Us is focusing on, they often refer to how you could learn more about a person by their zip code than you can by a lot of other things. And so All of Us is being very strategic about also making sure that every possible dimension of diversity is accounted for, including geographic diversity, socioeconomic diversity, ancestry diversity, et cetera, et cetera.
So interesting. And I'm going to ask one more of our questions from the chat. Sure. And then turn it right back to you. So Lana Skirball had a really interesting question. She posted a New York Times article.
Lana always asks really good questions, even from her
that is
back at NIH.
So true. That's the default position.
That's right. No other expectation.
I don't know if I had not seen this article. It has to do with genetic testing in pregnancy. So that she's asking about the analytic validity of prenatal testing, prenatal genetic testing.
Yeah, I just heard about this yesterday, and I'm not informed enough to make much comment. I think there was a New York Times article.
Yeah, that's what she's posted. Thank you, Lana, for that.
Yeah, I think it's a New York Times article, and I don't have enough information to be informed to know one way or the other. Certainly, this is something we need to be really cognizant of. My experience in general, and when it comes to genomics, is that the field has done incredibly well keeping very high data quality, but those are for research projects. And I think what this is talking about is, as this has moved into large-scale diagnostic arenas, whether some of the validity has kept up high standards enough. And so it's on my to-do list, is to find out what this article was and find out research. But again, I think once again, it's like anything else. When you transition from a research environment to an implementation environment, anything medical, we need to make sure that qualities are kept high, standards are kept high, and we don't want to shoot ourselves in the foot by not having missteps along the way. So
and the policy, the bioethics and policy context becomes so important. Anna, let me turn this back over to you. I know we have a lot of terrific questions.
We do, lots of terrific questions have come in. So Dr. Green, another question. This person is asking if you could offer any comments on the potential for genomics in treating mental health conditions, particularly through personalized medicine, and if there have been new developments in genomic testing in psychiatric diagnoses.
So I think there's an example where NHGRI works very closely with the Mental Health Institute. We do with all the institutes in their particular disease areas, and always asking, how can we be helpful? What can we do? Josh Gordon is the director of the National Institute of Mental Health and has invested considerable amounts of research dollars into the study of the genomic basis of mental health. I think everybody recognizes there's some clear genomic components or genetic components, but that's not the whole story. I think it is also but I think there's some very clear home runs, probably from, it must be about 5 or 6 years ago, where a disease as complicated as schizophrenia, for example, where, falls under one label. And we always had a hint that it probably was multiple diseases. And sure enough, and yet we know it's multiple genes. It's very complicated. And actually work that actually came out of the Broad Institute, the President Biden science advisor, Eric Lander, was a key leader in that study, where they actually teased out, they were able to get to one specific gene that clearly, when mutated, gave a much higher risk.
Wow.
Now, I give that as an example because it probably hasn't yielded anything quite yet, as far as I know, in terms of diagnostics, but it put them into a biological pathway they didn't know previously. So getting access to these pathways is really important. I think the other thing that we're hopeful for, but we're not quite there yet, is I think all of us recognize or know, either personally or from family members or friends, the odyssey of mental health medications, where it always seems to be like, you try A, and if A doesn't work, you try B, and if B doesn't work, you try C, and if C doesn't work, you try a higher dose. It doesn't seem very precise because it's not. And I think there's a lot of hope. It's still hope, though, that, this is this word pharmacogenomics, which is a combination of pharmacology and genomics. Are there genomic signatures, in other words, sort of patterns of DNA differences among certain people that would point them to either which is the best medication for them amongst a set of medications, or maybe what's the best dosing for them? And these are often DNA differences in drug metabolism pathways. And for some medications, this has proven to be outstanding. I mean, there's asthma medications, there's HIV medications, and some cardiovascular medications where pharmacogenomics is now being used. And we always keep hoping that, it just seems like for mental health, there should be that, because it seems that it's so arbitrary what works for somebody, what doesn't work. And wouldn't it be better if we could upfront figure out this might be one of these things where we just need bigger numbers. Maybe all of us, with its 1 million people, believe me, you look at 1 million people, you're going to have a lot of people who are going to carry in their medical records indications of what what mental health medications they've tried, which ones worked, which ones didn't. Maybe we'll get insights, and maybe we just need studies.
Oh, that's a really good point. Yeah.
So again, I think I wish I could. 10 years ago, I'd have said, "Oh, pharmacogenomics, it's going to hit home runs when it comes to mental health." We haven't seen that yet. We don't quite know why, but we haven't stopped trying, and I just think maybe we just need big enough numbers.
Interesting. So, boy, we've kept you over time here, but I know we do have a few more questions. If you could indulge us, Dr. Green.
Great.
Anna?
Sure. So Dr. Green, I believe this person said, in looking at the genome and environmental factors that can play in that space. Is there an intention or process where climate change will be factored in there or considered or looked at?
I'm being very hesitant because I would say, actually, I would tell you probably the answer is yes. It wouldn't be done by our institute. It's actually this institute, the National Institute of Environmental Health Sciences, NIEHS. That's the institute we're doing a workshop with next week. And certainly they, absolutely, that's part of what they do is environmental health. And they're working with us because we bring the genetics and genomics component. They're bringing that. So that's why we're looking for fertile areas of working together. I chuckled because I just want to point out that, and I have to be politically careful here, it is a, climate research is a tricky business because that's a political hot potato related to climate change. And so this is sort of, I know one of these areas where sometimes there's not uniform views of what NIH should and should not be doing across all the halls of Congress. And so, climate research is a tricky business. So, but it's not saying that we get directly involved in because we're the Genomics Institute, but I just point out it is fraught with some challenges on occasion.
Right.
Anna, there's a question in chat. I believe that you've touched on this, Dr. Green, but maybe I'm misreading the question. It says, does your institute only focus on human eukaryotic, excuse me for if I've mispronounced that, cells? And he asks about the Human Microbiome Project. It sounds like you were saying that it's so important to broaden the omics.
So let me put a fine point on this. So let's first make sure everybody knows what microbiome is. We now know that there are these communities of microbes, bacteria, yeast, viruses that live in us and on us. Where there's more microbe cells in our human body than there are human cells. It's just the reality, but we live in a very important equilibrium with them. Occasionally, when we get out of equilibrium with them, we get sick. And obviously, there's a lot of interest in individual infectious agents like coronavirus these days, but many others as well. Early on, our institute was very involved in microbiome research. We were the co-leaders of the NIH's Human Microbiome Project. Why is that? It's because genomics and the cheap DNA sequencing became a tool for being able to inventory microbes without ever growing them in the lab. So I could take I could rub a Q-tip on my skin. I couldn't grow 90% of the bacteria that live on my skin. I can't grow those in the laboratory, but I could purify DNA, read the DNA, and just have the computer tell me what bacteria are there. That's what microbiome research is all about now, is using DNA sequencing as a sidestep to culturing the bacteria, but you can get an inventory. It's like opening a window for the very first time and seeing one of the most intimate environmental influences in our life are the microbes that live in us and on us. This has caught fire, again, be blunt, because of cheap DNA sequencing. It has caught fire at NIH, so that our institute walked away from it for the most part a number of years ago because individual institutes picked it up to study it in the context of intestinal disease, in the context of diabetes, in the context of skin diseases, in the context of cancer. And so lots of institutes now do microbiome research, but the tool they use to do those studies is genome sequencing.
A terrific answer. And I think we'll stop there, but I just want to thank you personally, Dr. Green. I just think about how extraordinary your contributions are and in your leadership over this institute in revolutionizing medicine. And I think about rare diseases and the fact that we were nowhere. You know what I mean? That it was just a desert, and now there's so much hope. And so much of that lies in the work you've done and the work the Institute has done. So thank you broadly. And thank you specifically for being so gracious and generous with your time today.
And I'm delighted to share this. And again, I hope people use some of the electronic resources I introduced you to, because you go far deeper in anything I talked about. You can get far deeper into it, and we want you to take advantage of that. So
yes, and there's
thanks for
no
inviting
lack of
me,
interest.
and thanks to the folks who joined us today.
Well, it was our pleasure and our privilege. Thank you, Dr. Green. And I know I have housekeeping to do, but I'm going to turn to Anna because Anna, I need you to do the housekeeping.
No problem, Melanie.
I can't find my script. So
all good. So 2 quick announcements before everyone goes, links for which are being put into the chat. We hope you can join us right back here next week on Wednesday, January 12th, at 11:30 a.m. Eastern. Dr. Amira Roez, a professor of global health and epidemiology at George Mason's College of Health and Human Services, is going to join us to discuss what is known and unknown about the Omicron variant and its implications for the pandemic. The following week, on Wednesday the 19th at 1 o'clock Eastern, I hope you will also join us for a special discussion continuing our series highlighting our 2022 Advocacy Awards honorees. The awards are being held on March 16th, just a few months away, so get excited. For the special discussion, we will be joined by Sue Peshin, President and CEO of the Alliance for Aging Research, one of the most respected experts in the arena, of aging research and policy. Sue is going to speak with us about how the Alliance has evolved under her leadership, their priorities in 2022, and her vision for successful advocacy. So I hope to see you then, and for now, have a great rest of the day. Bye.